Good day, and thank you for joining us here at the Biopharmaceutical Summit. We are so excited to have you here for the next 30 minutes. We promise that we're going to share with you some very exciting information about how to increase efficiency in your biological workflows when you're provisioning nucleic acids. My name is Rhonda Petit, and I'm the Global Sales Director here at MicroGEM. And with me today is Dr. Georgi Hampton. She has a PhD in molecular microbiology, and she is our Chief Field Science Application Scientist. So what we'd like to go through today is on the agenda, we're going to talk about some of the challenges that we find in the biotherapeutic biopharma workflows and how we using a new concept and a new idea and letting how we can allow nature to do the heavy lifting in the nucleic acid provisioning steps. We'll give you an overview of our company, how this revolutionary approach works, the benefits, and then give you some use cases from an actual customer site where they really reduced speed, time, and increased their efficiencies in their production process pretty dramatically. And then we'll take some questions. So the only thing I ask as we start is that you allow yourself to accept a new idea that isolating nucleic acids actually can be easy. So the challenges that you hear about in all of the biological production facilities today really centers around three areas or three pillars that we like to talk about. One is speed. The second is utilizing process innovation, and the third is operational excellence. If you look at the cell cultivation market segment today, you'll find that we have, there are bioproduction development of things like vaccines, therapeutic proteins, and antibodies at a much higher rate than 20 years ago. There now are stem cell cultivations, tissue engineering, and you've got process workflows for RNA silencing and other cell-based assays like CRISPR. And versus 20 years ago, the focus really has been is going away from screening some compounds from a compound library and into screening a lot of cell lines. And with that, the processes of seeding, feeding, and hit picking and cloning become increasingly important in drug discovery. And in drug development, there's a lot of new opportunities for cell culture and cell line development, as well as automating those cell culture processes. And the problems and some of the challenges become around the maintaining the cell lines, eliminating reruns that can cause delays in time of getting the drug to market. And so we believe that what we're going to share with you today is going to give you some major opportunities to streamline those processes. Our company's name is MicroGEM, comes from the combination of two centers of excellence in terms of microfluidics and genes, enzymes, and microbes. We have an extremophile culture collection that was discovered in New Zealand and Antarctica, and that those enzymes really represent the strength of the product combination with qPCR miniaturization. As far as where we're located, and our company is in whole, we have global operations, R&D, and manufacturing across the globe. In Charlottesville, that is Virginia, that is our US distribution hub, as well as our expertise for the advanced microfluidics. In Dunedin, New Zealand, we have our Center of Excellence for R&D with our enzyme discovery, synthetic biology, and assay development. And in Guilin, China, we have manufacturing facility and R&D for China markets. And then over in the UK, our finance and legal operations are located there with our European distribution hub. The vision of the company was really to democratize molecular biology and bring it to point of care so that you can have molecular performance at the point of need. So we're using novel approaches, uncompromised test speed to really bring the precision medicine where it's needed. So some examples are, we've had our technology taken into the fields in Tanzania for detecting Cassava mosaic virus in a farmer's field. Our technology has been used in the ocean to detect pathogens. And most recently, our technology is being taken into help with the COVID-19 tests that we are introducing that will go in places such as dental offices and where it's needed at point of care. So the whole idea is, how do you bring complex molecular biology techniques to the point of need where they can be used by anyone, anywhere? And so the key to the company is, and the technology is really this temperature-driven DNA and RNA extraction. We let nature do the work. So the enzyme is going to do the work to isolate the nucleic acid without all the washing steps and do it in a single tube approach. So the sensitivity and speed are going to be realized. There's much less hands-on time steps in the process. And from an efficiency standpoint, there is decreased exposure to any kind of hazardous materials and extra steps in the process that can cause concerns for contamination. The microfluidics simplifies the processes with miniaturizing the sample prep extractions and allowing for qPCR, the gold standard, in a single system. As an example, we have been awarded, due to this technology, the NIH has a RADIX initiative, and we were awarded a $42 million grant to bring a testing system to market that is going to be able to detect SARS-CoV-2, influenza A, and influenza B in one point of need lab test, again, for anyone, anywhere where it's needed. As far as our operations globally, we're building in operational excellence with our innovative products and our commitment to quality, safety, leadership, and employee engagement, strategic direction and focus, and process excellence. And we expect to have our ISO 13485 quality policy in place by the end of Q1 of 2021. And at this time, what I'd like to do is turn it over to Dr. Georgi Hampton, who will explain to you our revolutionary approach to nucleic acid provisioning. Georgi?
Thank you, Rhonda. Next slide, please. So conventional nucleic acid extraction workflows typically involve one of the following: either phenylchloroform extraction, or you'll use a silica-based column, or you might bind your DNA and RNA to magnetic beads. And all these processes essentially have a cell lysis step and then an extraction step. Then after the extraction, you have to remove the harsh chemicals that you've used in the extraction and wash with ethanol, then remove the ethanol, and then finally you're left with your DNA ready to be used in downstream applications. Next slide. So instead, what we do for MicroGEM is we utilize enzymes and temperature. So it's a much simpler process. We have our enzyme, buffer, water, and sample into a tube. Then you just heat it up, and then after that, your DNA or RNA is ready. So what happens is our enzyme is a thermophilic proteinase, so it works at high temperatures. Its optimal activity temperature is 75 degrees Celsius. So you incubate for about 10 minutes at 75 degrees, then have a short incubation of about 2 minutes at 95 degrees, which denatures the enzyme, and then you're left with DNA ready to be used in whatever you want to do downstream. Next slide. So we cut out all of the traditional steps of a traditional extraction process, using our enzyme and the temperature-driven extraction approach, you can cut down your extraction from using 5 to 7 steps to just 3 steps, and to be able to achieve a full extraction in under 20 minutes. So this MicroGEM's extraction process is also carried out in a single tube, which has a wide range of advantages. Not only are we fast, as I just mentioned, but because we don't wash the nucleic acid, you get 100% recovery. Whatever you put in the tube is what you'll extract DNA from, and there's no wash steps where you might lose some of your precious nucleic acid sample. We also don't have any need for harsh chemical washes. The fact that you're working in a single tube and not opening that tube multiple times or changing the tubes means that you reduce the chance of cross-contamination and contamination from the environment, as well as human error, because there's less labour steps involved. We also produce substantially less environmental waste, like chemicals and plastics, than competitive techniques. We also have a wide range of buffer systems that are capable of extracting DNA and RNA from even more challenging samples like plant samples or bacteria samples. Next slide. So not only are we shorter in time, we're also, the steps that are involved in a MicroGEM extraction contain a lot less labour than traditional extraction methods. So the phenochloroform columns or magnetic bead extraction processes, a lot of those steps require technician labour and time, whereas MicroGEM, you just put all of the things in the tube, put it in a thermocycler and walk away. So as you can imagine, this is a very easy process to automate. It's so simple that you can easily program a robot to do it for you, which reduces contamination even further. And we can easily scale these extractions to work on hundreds of samples at a time by carrying out extractions in a 96 or 384 well plate. Slide. So we've also worked with a few liquid handler automation companies to work on solutions to automate the entire process. So there's ways that you can perform the extraction and qPCR setup of your downstream application using a robot. So that means that you don't have to have technicians do anything apart from initially set up the system. And that means that you can get up to 4,600 extractions a day per system. And if you need more, then you can always scale with additional systems. But the fact that the process is so simple allows you to maximally utilize your capital assets like liquid handling robot systems. So not only that, we also tend to see that MicroGEM's processes have optimal recovery of nucleic acid compared to other extraction methods. So this is comparing MicroGEM extraction in blue to a competitor extraction in grey, and you can see that at every dilution performed, you're getting a lot more DNA yield out of the extraction than with competitive techniques. And the main reason for this is because we extract in a single tube and don't do any wash steps where you often lose DNA. So this schematic is just showing all of the chances for contamination that traditional methods involve. So the black triangles show every time that the method requires that you open tubes. So as you can see, a prepGEM universal approach only opens the tube 3 times compared to other extraction methods where you're opening the tube a lot more. So that means that we have reduced contamination, and we also see that the blue steps are steps where it's a hands-free step. We also have a lot less labour-intensive steps compared to traditional methods. So we have our extraction enzyme, but that's just one enzyme out of potentially hundreds of thousands more that we have. So our enzyme was discovered from our culture collection, which is a big range of bacteria and fungi that were isolated from Antarctica. These organisms, next slide, please. These organisms have a physiological temperature range from -40 to 105 degrees Celsius. So they're almost all of them are extremophiles living in either very cold or very hot environments. And in addition, they've got a vast range of pHs. So we have a couple of screening libraries of purified, well-characterized molecules, as well as crude fractions. And as you can imagine, this is just a treasure trove of discovery for potential novel enzymes, new molecular targets, and new antibiotics. So just one example on the plate that you can see on the right that is growing a lawn of multi-drug resistant Staph aureus. And you can see 10 fungal strains that have been seeded onto, that are growing on top of that. And we can see that there are 3 penicillin-like isolates in the top part that clearly secrete some kind of antimicrobial that are capable of killing this problematic strain. So we have some interesting things that are just waiting to be discovered. Next slide. So our current products center around prepGEM, our enzyme that extracts nucleic acids. We have kits that are designed for normal tissue, cell culture. We have kits more centered for RNA. We have plant kits, and we also have forensic kits. We provide our products as chemistry only, where you can use your own thermal incubator, or we also have a pqEx nucleic acid extractor, which is a instrument that we produce also, which incubates tubes at specific temperatures, but it also uses a specialized consumable that allows us to get double-stranded DNA out of a single tube extraction that is suitable for some next generation sequencing approaches. So we serve a vast range of markets. Our customers come from a wide range of applications and laboratories, ranging from food, agriculture, and environment. So a lot of environmental monitoring, molecular forensics, so that we have people that use our products for analyzing rape kits, biopharmaceutical R&D, biomedical research, also people using our products in precision medicine and for point of care diagnostics. So one example that I'm going to give you, there's a vast range of ways which MicroGEM's extraction chemistry could be useful in a biopharmaceutical workflow. But one example is in CRISPR Cas9 workflow. So I'll introduce that as an example. So just briefly, a CRISPR Cas9 system is a microbial adaptive immune system that uses RNA-guided nucleases to cleave foreign genetic elements. Once that foreign genetic element is cleaved, it is then destroyed by the cells, so it works as an immune system. But almost as soon as this was discovered, researchers realized that this could be potentially a genome editing system for eukaryotic cells. So this had great significance because suddenly we could think about the possibility of engineering biological systems, repairing mutations, designing new ways to target mutations and repair them. And it has enormous potential for applications across science, medicine, and biotechnology. Next slide. So overall, a CRISPR workflow to establish a knock-in or knockout mammalian cell line is you start with your in silico design of the primers and vectors needed. You then have a few days of molecular cloning, followed by transfection, and then the functional validation of the short guide RNA, so checking that your RNA constructs are doing what they should be doing. Following this, you have clonal expansion of your clones that have the correct gene editing that you require, and subsequently single clone screening. So both steps where MicroGEM's products can save you a lot of time in this process are the functional validation of the short guide RNAs, as well as the single clone screening. So most of the time taken up by a CRISPR genotyping workflow involves the time taken to grow the cells. Often eukaryotic cells are a bit slow to grow compared to bacteria, and traditional DNA extraction techniques require a large number of cells as a starting material because of that DNA loss throughout the method. That means you have to grow a large number of cells in order to do an extraction. But with MicroGEM's extraction chemistry, because we're able to scale to any volume, you can perform genotyping at a much earlier stage. You don't need to grow at nearly as many cells before genotyping. Next slide. So using prepGEM Universal in the screening process and the later confirmation of your clonal lines process means that you can save a lot of time, up to 2 or 3 weeks in the process, simply because you can get 100% DNA recovery out of the cells from a really small starting volume. And next slide. This was successfully used by some of our customers. They simply took a cell pellet containing about 5,000 to 20,000 cells, incubated that in a 96-well plate with some prepGEM and some blue buffer, incubated at 5 minutes for 75, incubated at 75 degrees for 5 minutes, and then 95 degrees for 2 minutes. And they end up with a plate of single-stranded DNA that's ready to be used in a conventional endpoint PCR to determine the genotype. And they then were able to sequence the PCR products to confirm the genotype successfully. So they managed to successfully genotype after only 10 days of clonal expansion, and usually this would require 3 to 4 weeks. So when you're cutting down the time required to grow your cells to confirm the genotype, not only are you cutting down on technician time required to passage and seed and feed those cells, but you're also cutting down on the space required to grow the cells and the media, consumables. You can save a lot. Slide. So the applications when you use MicroGEM in these CRISPR workflows is wide ranging. There are so many different things that you can do with CRISPR editing. You can make cell lines, knock in or knock out genes, make targets that knock down expression of genes. You can edit bases in your cell line. You can also get into CRISPR interference and other techniques. The possibilities are really endless.
Thank you, Dr. Hampton. We really believe that the possibilities are endless specifically in the biopharma workflow process, because the nucleic acids for all these in all the cell cultivation that's going on play important roles in the function and the performance of the cell lines to the end of making, whether it's vaccines, therapeutics, we have doing any kind of cell screening with stem cells or silencing. There's a whole culture of drug targets that we have access to that could be used. And certainly from a companion diagnostic standpoint, either whether it be prognostic screening or a point of care, we can see that the technology could be used with these cell culture workflows to really increase efficiency, drive the speed of the drug to the market. You know, if you're looking at going to getting something done in 10 days versus 3 to 4 weeks, think of the impact that that can make on getting the drug to market faster. This cost savings it can provide from an efficiency standpoint. So whether you're in research, working with biological markers, or any kind of research diagnostics, or drug discovery with all the assay development and screening, or in the production of drugs that are requiring, you know, the nucleic acid, whether it be a DNA or an mRNA vaccine production, these workflows could apply, as well as a companion diagnostic. So really, again, at the beginning of this, I asked you to accept the idea that nucleic acid isolation can be easy. We've traditionally used columns and magnetic beads, and today now there's a natural way to go about isolating those nucleic acids to get higher recoveries at a faster rate with less effort, and less headaches. So single tube workflow, temperature driven extractions, not a lot in the process. It's fast, it's economical, it's reducing contamination, eliminating human error because there's less human steps, in reduction of plastics and consumables. So at this time, what we'd like to do is take any questions you may have. And again, thank you so much for taking the time to find out about our products. We encourage you to visit our website or reach out to contact us if you have any additional questions. But at this time, we're going to take live questions. Thank you.